Hi everyone, thank you for watching. My thesis is titled Secondary Metabolite Regulation in Streptomyces silicolor via the MIBO Biosynthetic Cluster. My name is Samuel Jewett and I've conducted this work under the mentorship of Dr. Taifo Mahmood in the College of Pharmacy. So Streptomyces species are famous for producing the majority of our clinically used antibiotics. But in their natural soil environments, these secondary metabolites don't just function as antibiotics. They also function as ecological signals that can help bacteria compete, communicate, and even respond to stress. And so because producing these molecules is actually extremely energy intensive, their biosynthesis is pretty tightly controlled. Streptomyces uses several different layers of regulation, including global regulators and pathway-specific regulators to turn metabolic pathways on and off depending on different environmental cues. And so our model organism for studying these systems is Streptomyces silicolor. Its genome contains many biosynthetic gene clusters, but under laboratory conditions, a large number of them actually remain silent or cryptic. And these silent clusters are especially interesting because they might encode unknown metabolisms or even hidden regulatory systems. And so one of the clusters of interest is the MIBO locus. And this region resembles the EBO and EBD clusters found in gram-negative bacteria that the Mahmood lab had already researched in the past few years. And these clusters regulated the production of small signaling molecules such as indole derivatives. And so a key feature of the MIBO cluster is that it contains a MIPS-like gene. And a traditional MIPS-like enzyme, myo-inositol-1 phosphate synthase, catalyzes the first committed step in inositol biosynthesis. And inositol derivatives in bacteria usually serve as signaling molecules, which influence stress responses or metabolic regulation. And so the version found in MIBO is MIPS-like, meaning it is homologous but not identical. And these MIPS-like enzymes often produce inositol-derived intermediates rather than canonical inositol. And in many organisms, these intermediates actually act as regulatory small molecules, modulating transcription factors or interacting with other metabolic control systems. And so because the MIBO cluster encodes this MIPS-like enzyme and also lies outside any major biosynthetic pathways, we hypothesized that it does not function primarily as a standard biosynthetic cluster. Instead, we predicted that MIBO acts as a regulatory locus, potentially influencing the production of actinorodin, a well-known antibiotic that's produced by Streptomyces silicolor or other metabolites. And so the main research goal was to determine whether the MIBO cluster acts as a regulatory locus in Streptomyces silicolor. And more specifically, our hypothesis was that the MIBO cluster modulates secondary metabolism with a particular impact on that actinorodin production, which produces a strong blue pigment that the species is known for. And so to test this, we compared three main strains: the wild-type strain, the delta SCO6573 deletion mutant, where the key gene in the MIBO cluster was removed, and a complemented mutant where the gene was restored on a plasmid. And across these strains, we used a combination of approaches to measure changes in metabolic output. This included phenotypic observation of pigmentation, UV absorbance assays to quantify total metabolite levels, LC-MS to identify specific compounds, and HPLC to generate detailed metabolic profiles. And so together, these comparisons allowed us to really directly test whether the MIBO cluster influences that actinorodin antibiotic production. For this project, we first generated a delta SCO6573 deletion mutant using what's called a standard double crossover strategy. And then we verified all constructs through gel electrophoresis and PCR to ensure that the genotype was correct. And all the strains, the wild type, the deletion mutant, and the complemented strain, would then grow under identical conditions in BTT medium. And this was mainly so that any difference in metabolite production could be attributed specifically to the loss of SCO6573 and not any environmental factors. And then to analyze metabolic output, we then extracted these metabolites using ethyl acetate, which essentially pulls out the major antibiotics and signaling molecules that are produced by Streptomyces. We quantified and characterized these metabolites using a multi-step analytical approach, UV spectrometry at 640 nanometers to measure total actinorodin-like metabolites. We also used LC-MS to identify the specific compounds, HPLC to provide a fine scale separation, and also to visualize differences across the full metabolic spectrum. And then together, these methods really allowed us to assess both the quantity and identity of the metabolites impacted by the deletion of SCO6573. So the results, when we look at the phenotypes of each strain, we see a pretty clear difference. As you can see, the wild type strain producing that deep blue pigment, which is the main indicator of actinorodin production, and that kind of served as our phenotypic baseline. And then you can see the delta SCO6573 mutant showed a dramatic loss of that pigmentation, which shows, or at least it suggests, that removing SCO6573 reduces that actinorodin synthesis. And then also, very importantly, we can see when we introduce the plasmid back, you get that blue pigmentation being restored. And so this rescue really suggests that phenotype isn't due to any off-target effect, is really directly reflected in the function of SCO6573. That's in B. And then in A is just showing you that gel electrophoresis that's confirming the loss of SCO6573 in the mutant. Moving forward, UV spectrometry. To quantify the overall metabolite production, we used UV spectrometry at 640 nanometers, which is commonly used to measure actino-like compounds. And we can see at day 4, the wild-type strain showed an absorbance of about 1.79, while the knockout mutant only showed 0.52. And so that's just about a 3.5-fold decrease in the mutant in terms of secondary metabolite production. But then by day 5, that became even more dramatic. You see the wild-type increased to 8.21 absorbance units, whereas the mutant only reached 0.81. Which shows more than a 10-fold reduction relative to wild type. And so these measurements really show that the mutant not only produces fewer metabolites overall, but it also fails to undergo the typical late-stage metabolic burst, which really, again, supports that conclusion that SCO6573 plays a major role in controlling the timing and magnitude of secondary metabolite production. So then moving forward, HPLC analysis gave us a little bit of a more detailed look at individual metabolites. In the wild-type chromatograms, we saw pretty strong, well-defined peaks, including prominent actinorodin peak. In contrast, the delta knockout mutant showed a dramatic reduction in peak intensity, specifically in that actinorodin region. You can see that in peak 7. And but more importantly, what was really interesting is that the mutant didn't just lose actinorodin, it also lost additional peaks of unknown identity, and they were also noticeably altered. Some were reduced, others shifted in intensity, which means that the mutant, the SCO6573, might not just be regulating one pathway. It could be that the MIBO cluster influences a broader set of metabolites. So it really might it really reinforces this idea that it acts as a regulatory locus rather than a traditional biosynthetic cluster. Next, we use LC-MS to look at specific metabolites with much higher resolution. In the mutant, the ions associated with actinorodin were strongly reduced, which is fully consistent with that loss of blue pigmentation and drop of absorbance we saw earlier. Interestingly, we were looking at other metabolites and how they might be affected, including undecylproteoglycosan, hopine, and germicide A. And they showed little to no change between the wild type and the mutant, which is pretty interesting. And so while the SCO6573 clearly has a major effect on actinorodin production, it doesn't seem to suppress those other metabolites. Overall, our findings support the idea that the MIBO cluster is functioning as a regulatory locus rather than a traditional biosynthetic cluster. And this mirrors what has been observed in Pseudomonas fluorescens, where the EDB cluster regulates the production of small signaling molecules. And one possible mechanism for this might be that the inositol-derived intermediates produced by the MIPS-like enzyme in MIBO acts as signaling molecules. And these intermediates might interact with key regulatory proteins in streptomyces silicolor, such as ACT2 or F4, which activates actinorodin production, or with broader global regulators such as SCBR and SCBA. A really interesting aspect of MIBO is that it exists in two genomic locations. So one copy is on the chromosome and one is on the plasmid. And we think that these two copies may serve slightly different roles. The chromosomal copy might provide more stable developmentally programmed regulation, whereas the plasmid copy might provide more dynamic, environmentally responsive control. And again, this kind of layered regulatory architecture would give streptomyces greater flexibility and help it compete in consistently changing conditions of soil environments. Stepping back, this work contributes to a larger shift in how we think about cryptic gene clusters in streptomyces. Traditionally, these clusters were viewed mainly as silent biosynthetic units. They were essentially potential antibiotic factories waiting to be activated. But what we're seeing here is that some of these cryptic regions may instead function as regulators, shaping more when and how metabolites are produced. And so understand these regulatory clusters opens several exciting possibilities. We might be able to unlock silent biosynthetic pathways that don't normally activate in the lab. We can also potentially improve antibiotic yield by tuning regulatory circuits rather than altering biosynthetic genes directly. And this expands our ability to discover new natural products, especially those hidden behind uncharacterized regulatory switches. And ultimately, studies like this really help us start to decode the broader regulatory logic of streptomyces, a logic that underlies the enormous chemical diversity that this genus is capable of. So to summarize, deleting SCO6573 caused a strong reduction in actinorodent production and also altered several additional metabolites. And this tells us that the MIBO region isn't acting as a traditional biosynthetic cluster. Instead, it behaves more as a regulatory locus that helps shape secondary metabolism. And the dual localization of the cluster on both the chromosome and the plasmid suggests a more sophisticated layered regulatory system. And then finally, clusters like MIBO highlight an exciting opportunity. Regulatory loci might be powerful levers for activating silent biosynthetic gene clusters and also expanding natural product discovery. And by understanding these regulatory networks, we can better unlock the metabolic potential of streptomyces and improve access to novel antibiotics. Thank you all for your time. I really appreciate it.